tell I'm still having technical problems, so I apologise for that, and we'll try to make this video as short as possible for that reason, and others. So I may get cut off again. So I just wanted to say, before we start going ahead to look at meiosis, we have one more topic to cover on DNA replication, and that's looking at DNA replication in the laboratory, so outside of the body, in terms of biotechnology. But before we go there, in the last lesson we looked at how RNA, another nucleic acid, is involved in replicating the template of DNA and ultimately in protein synthesis. So the DNA template, remember, is copied by messenger RNA and coded into a separate code, which is taken as a triplet codon to tRNA in the cytoplasm near the ribosome. The transfer RNA then produces an anticodon to that codon brought by the mRNA, which in turn designates a specific amino acid. So each codon that's brought is for one specific type of amino acid, which is then joined together in a long chain to make a protein. So that's what we looked at last time. I'm aware that I rushed through that because of these technical problems that I'm having. So if you want me to go through it again, let me know, and I'll do a longer video once everything is fixed. Because this is an important topic, protein synthesis will come up in the exam, and quite often there's an essay question on it as well. So I'm perfectly happy to make another video on that one. As yet, we don't know when your exams are going to be, so that's helpful in some ways. It's not clear. We know the curriculum is being altered this year, but again, we don't know how. And now there's suggestions that it may be cancelled altogether. So we just don't know yet. The government hasn't provided details, but it's better that we keep you informed, and then at least you'll be prepared for the exam whenever it happens, and you'll be prepared when you go back to school. So I hope it's still useful, even if we don't end up sitting exams this year. So as well as looking at how protein synthesis last time, we also focused on the part of the DNA molecule which is non-coding. So the non-coding DNA, otherwise known as short tandem repeats. And this is used in solving crime, so in forensics, in building up a genetic fingerprint, and in genetic profiling. So if you missed that, have a look at lesson number 2, and it's in there. I have split up the video into several parts because of this problem I'm having. But I promise to fix it as soon as possible, because it's driving me as mad as it probably is you. So that will come up as well. That's likely to come up, especially the patterns, the banding. If you're trying to work out, match a suspect of a crime with the victim, that's likely to come up. And I had a diagram which you would be able to see better last time. I apologise for the lighting today as well. But before we go on to look at these techniques used in the laboratory with DNA replication, I wanted just to cover again these two important words which are associated with all types of DNA replication, so whether internally in our bodies or in the laboratory. So it's very important, remember, that you know the difference between these two words, transcription and translation. So transcription is the process where the mRNA, messenger RNA, converts the DNA template into a coded message. So into the coded message of messenger RNA. And it then carries it to the ribosome where proteins are synthesized. So that's transcription. So you can think of it as a transcribed message from the original DNA template into the messenger RNA, carrying it as a coded message. So that is the transcription. And the translation, on the other hand, is translating the original DNA template into a specific amino acid to be joined to the protein chain. So the process of linking specific amino acids together according to the anticodons of tRNA, transfer RNA. So it's actually the process